Ladies and gentlemen, Happy New Year on 29th of January. We are happy to be back. My name is Dr. Thuranira Kaugiria from Kenyatta National Hospital. Today we have a full schedule. We have several presenters, 3 to be specific. We have a guest presenter from Kemri Welcome Trust. So I wanted to do a quick round of introduction to Salimiano. Dr. Marybeth, show us your face, say hi to us.
Happy New Year, everybody. Good morning. I'm happy to be here. Thank you.
Thank you. Remotely, Dr. Nyukuri.
Good morning, everyone.
Our guest, Charles.
Good morning, everyone.
Start your video.
I'm not allowed to switch the video on.
Okay, we'll sort out that one shortly. And our boss, Dr. Life.
Good morning, everybody, and Happy New Year. It's good to see you today. We have an interesting discussion lined up today, focusing on the issue of variants of concern for COVID-19, which is something I think that all of us have been observing and wondering about and wondering exactly what that means to us. So we'll go ahead and start. I'll just share a couple of slides before we start. So everybody has been asking the question of, have we flattened the curve? Is COVID gone? Is COVID still here? What's happening? And based on the data that's been shared, at least globally, we know that there's still a huge concern with COVID, especially with the onset of these variants of concern. If we look at this map, looking at daily new cases daily, and this is as of yesterday, we see there's some regions that are still reporting close to 100,000 cases per day, like the US. Brazil has seen a recent very large spike, reporting more than 50,000 cases a day, and some of the European countries still remain quite a big concern. And locally, South Africa has also reported close to 10,000 cases a day. So this is something that we have to keep watching closely. And I think with infections such as COVID, or indeed any other infectious disease, one region is safe only when all the regions are safe. So as long as there are areas that are still struggling, we cannot afford to put our guard down, because this could very well be us at some point. If you look again, I just mentioned this, some of the countries that are really reporting a large number of cases. Brazil is a case in point because, again, some of the regions that are reporting very high cases have actually run out of, the capacity to take care of patients. So you have patients dying just because they cannot access oxygen. So for every region, it's so important that they do not overwhelm their health capacity, just so that we do not increase mortality. As I said, South Africa has been a significant concern. As you can see there, up to about more than 8,000 cases in just one day. And so at home, we have not seen much of a spike. So if you remember, November was our largest sort of case reported, as well as mortalities. Again, the initial expectation was that after all the December festivities, and after the school reopening, we may begin to see a few more cases. I'm sure for the hospitals that are taking care of patients, we've seen a slight creep in numbers. So from a low maybe of just 10 cases in our hospital sometime in December, to this point where we probably just have 25. So it's a small creep, and it's something that we have to keep an eye on, to keep vigilant, so that we do not miss patients coming into the clinical areas, especially the general medical ward, so that we don't have outbreaks within the hospital. Deaths have also remained fairly low, especially this month, but a cumulative of 1,753 deaths, as summarized by the Emergency Operations Center. If we look at some of the specific counties, again, this is data summarized by the Emergency Operations Center. All the counties are reporting a decline in cases. So Mombasa had one of the biggest peaks outside of Nairobi, but we've seen a decline. Of course, there's a concern of some of these variants from the UK and South Africa having been identified. And the question is really how well we can identify the contacts, isolate or quarantine and test so that we avoid any undetected spread in the area. And the same sort of applies to all the other regions. These are sort of the top 10 affected counties. So Mombasa, Kiambu, Nakuru, and the rest. And all these have seen a significant decline in cases. So of course, the question on everybody's mind is, now that we've been told there was some variant detected in Kenya, what does it mean for us? What are these variants of concern? What should we be looking out for? And today we have no one better to present to us than Dr. Charles Nyagoti. Dr. Charles Nyagoti is a researcher from Kemri Wellcome Trust who's been doing a lot of work around the genetics of COVID-19, and they've tracked the variants of COVID-19 that have been in the country. And he'll present to us what this is. And after that, we'll just have a brief discussion on what this means for us and what we can do to prevent an upsurge of COVID-19, of some of these variants of concern in the country. So I think I'll just stop there and invite Dr. Nyagoti to give us his presentation. Thank you so much for making time to join us, Dr. Nyagoti. We appreciate your very busy schedule, and we're very grateful you could find time to join us.
Thank you very much, Dr. Lois and Thuranila, for that kind introduction. And I have to say it's a privilege for me to be able to join this meeting to give you an update about the variants of concern. So I'm going to share my screen to allow me begin the presentation. Can I request for permission to share screen?
Are you able to share now? Just check again, please.
Let me try again. Yep.
Okay.
Are you able to see my screen?
Yes, we can. Thank you.
Okay, just a minute. I like to stop the video because need more bundles to be able to see the slides better.
That's
so.
Okay.
So I'm Dr. Charles Agoti, work at the Kemri program in Kilifi. Kemri program in Kilifi is one of the centers of Kemri, which has headquarters in Nairobi. And Kilifi actually is a coastal town. I'm sure quite a number of us in the meeting have visited coastal Kenya. So in this rural bit, we have the Kemri Wellcome Trust program, which is just by the coastline actually. So work on respiratory viruses started here at the Kemri Wellcome program about 20 years ago, when we started looking at pneumonia, in particularly viral pneumonia, with specific interest to respiratory syncytial virus. And eventually that has grown to encompass other respiratory viruses, including influenza, metapneumovirus, endemic coronavirus, etc. And it's really important also to say that this work which we'll be doing in the coast of Kenya is one which got my training both at the MSc and the PhD level into the molecular genetics of viruses, and especially encompassing genomics to understand various aspects of viral transmission. So it's because of that experience that we've had at the institute, and also at my personal level, having training that level, that I was able to quickly engage myself in responding to the COVID pandemic. And right from the beginning at the Kemri Kilifi program, we were able to set up the diagnostics for the coastal region for SARS-CoV-2, and quickly were able also to set up the genomics work for responding to the SARS-CoV-2 pandemic. Just to give you a little now specific about the SARS-CoV-2 virus, as a molecular virologist, when I think about the virus, I think much more of what you're seeing on the screen there, unlike maybe clinicians who might be thinking about a patient who is struggling to breathe in a ward. But what's this virus SARS-CoV-2 actually? It's an RNA virus. RNA meaning that its genetic material is made from ribonucleic acid, unlike maybe human genetic material, which is coded in deoxyribonucleic acid. And that RNA, which is the genome of the virus, about 30,000 nucleotides. And those 30,000 nucleotides encode proteins that make the virus structure and make the virus function in terms of being able to replicate and being able to transmit. So importantly, on the virus structure there are this protein called the S, which is what is found on the surface of the virus, making giving its name the corona, which is kind of the decoration on the surface when it was first looked at the microscope. But also there are other proteins like the matrix and the nucleocapsid and the envelope, which make part of the virus structure. You can see them there. Of particular interest is the spike protein, because this spike protein is the one which actually allows the virus to bind to our receptors, the so-called the ACE2 receptor, the angiotensin converting enzyme, the receptor that the virus binds, and that is for the spike protein. And also of interest that now that spike protein, because of its exposure to our immune system, it's been the one which is mostly targeted by our immune responses, and vaccines are exploiting that to actually make a virus which is targeting this, vaccine which are targeting mostly the spike protein. And we're talking about the Moderna vaccine, the Pfizer vaccine, and even the AstraZeneca vaccine. Of course, the other vaccines which target the whole virus when they have inactivated it. It's important to also know about the structure of the spike protein, which have kind of given there its full length. Importantly is that it's made up of domains. There's the S1 domain and the S2 domain. And once the virus changes this nucleic acid sequence into the protein, which you're seeing there, the structure is sort of what you are seeing here. Some people call it like the shoulder kind of, bent shoulder kind of. And not the parts of the virus that interact with the host cell are this part here we call the receptor binding domain. And when you look at the entire length of the spike protein, it's about 1,300 amino acids. The receptor binding domain is there, and changes which affect this receptor binding domain, which is given there, tend to actually change the way the virus interacts with the receptor. And that's why some of the changes are being quickly flagged as of concern. There are other parts, for instance, immunologically, the N-terminus domain is important, and also the receptor binding domain. But also importantly is that these two are also being used in serosurveillance. So changes occurring there are of interest in terms of looking at a range of our other assays, including the serosurveillance and the molecular assays, which might stop working if we are having changes on the spike protein. So there's this talk always that I've seen a hand raised. Shall I take questions during the talk?
No, we won't have any questions. The questions will be typed in the Q&A session, and we can have it at the end of your talk.
Thank you very much, Lois.
Thank you.
So in terms of thinking about the virus, there is always this talk, oh, coronavirus is mutating. I have to say, actually, as a virologist, viruses are always mutating. It's their nature. And if you, for instance, take here a graphic where we've shown the various SARS-CoV-2 genomes, you're looking at the genomes which were collected at the beginning of the outbreak in January 2020, and you're looking at the isolates you're seeing at the moment, and we are plotting here nucleotide differences against time. And as you can see, as time goes, the nucleotide differences from the original strain are increasing in a clockwise manner. Kind of every month, you expect to have about 2 nucleotide changes on the genome. These are not because they are advantageous sometimes, just because the virus replicates using an error-prone mechanism, and some of those errors are kept. They are sometimes not detrimental, sometimes they are removed from the system because transmission might not occur. But also another important thing that can occur during the evolution of SARS-CoV-2 is what you call the sudden changes in the genome. For instance, you can see here, when the molecular clock is saying perhaps this, we need to have changes around there, you're seeing some viruses which are quite distinct and they are varying, they are moving away from the expected line. And that's what you call the sudden jumps in changes. And that's an example, for instance, what can be observed in people who are immunocompromised, who get infected for so long. And during that, because the replication goes on and on, they end up incurring more mutations, and some of those mutations can be kept. But also, if you have two genetically distinct strains co-infecting an individual, you can have something called viral recombination, whereby two viruses which are slightly distinct recombine, and then you end up with a much more distinct virus. So if you look at the outbreak genomes at the beginning of the outbreak, for instance, here, the virus which we had circulating in January 2020, you can see they have very few genetic changes. This kind of, that's one nucleotide change, that's one nucleotide change there. So most of them hardly had two nucleotide changes or more. Most of them were either identical or just one nucleotide change. But by May, you already have viruses which are quite distinct from each other. You can see the scale has changed here now, it's up to 5, it's much smaller. And you have already now gotten so many variants just because of the natural process of this, of the nature of these viruses. And just to also give an example, is that coronavirus actually is one of the fairly low mutating virus. Flu mutates at twice the rate of coronavirus, and HIV mutates at 4 times the rate of coronavirus. So it's really important to just start putting this into the context. And because of this kind of mutations we are seeing, it's important for researchers and scientists to be able to track the virus it circulates in the population, and what the changes or the continuous change in the virus is helping is actually helping you know, if you find two viruses which are identical, it's unlikely they diverged 5 months ago. It's likely they diverged maybe 1 month ago or less than a month ago, because you said you expect 2 changes for every month. So because of that, over time now, if you look at this, what you call the current global phylogenetic tree of coronavirus, you can already start seeing we have now very many clades which have been named already by some groups. For instance, the next strain group, which is based in the US, has already named the current circulating coronavirus into 9 clades. And important to say the variants of concern, you can see them there. That's one of them is called variant 1 there and variant 2, but I'll say more of that shortly. But another system of naming tries to go into much more detail, much more of the variation that you are seeing even within a clade, and that's what you call the pango lineages. And for the pango lineages, they have identified up to 500 different variants, and actually that's a conservative estimate because sequencing of coronavirus is only at the moment about 500 genomes or thereabout, and we know cases are already 100 million. So if you expect, we sequence much more, we'll expect to see much more. And the naming system is important to say that for the pangolin, it's trying to use for B1 and B.1.2. It means that actually B.1.2 has some changes shared with B1, only that it has some unique change which has been allowed it to be given the next level. And you can see here B.1.1.119 has some changes which has made it to move, at least there are some changes which are unique to it down the line. I won't go to the details of the naming system today. So variants of concern are the reason why we are I'm with you here today. It's just to really define what are these. They are variants which are showing epidemiological, immunological, pathogenic properties that are worrying. And what do we mean by that? Epidemiological, for instance, if you are seeing a particular strain which seems to be spreading very fast in a location, then there is a danger that it might actually overwhelm the healthcare system. You remember flattening the curve. So it's making very hard for the idea of flattening the curve to happen. So it becomes what it becomes a variant of concern, or a variant which is being shown to have propensity to reinfect. We know there have been documented cases of reinfection, although they are isolated, they are few. But if we find any strain which is showing more frequently in reinfections, then that's a variant of concern. Of course, we know we have now some treatments, for instance, the monoclonal antibodies, the drugs, they are not there really, but the monoclonal antibody, you remember Regeneron, for instance. So these are monoclonal antibodies target specific areas of the virus genome. So if anything happens that makes those antibodies not to work, it's a variant of concern. Of course, we know we have vaccines and everybody's worrying what will happen to the vaccines if we are having variants which are escaping immunity. And then lastly, if we have a variant which we think is causing more severe disease than other variants, and then that becomes a variant of concern. So there are currently 3 global variants of concern. One of them is called the B.1.17, B.1.351, and B.1. So the first one is called variant 1, let's call it today. The second one is called variant 2, the other one is called variant 3. Variant 1 was first identified in the UK in September. Spread really quickly, because at the moment you've seen it in more than 60 countries. And actually this data as of 26th of January, I suspect it's been seen in more countries. And if you remember very well, when Corona came into existence, the first month we had it was in China. Everybody thought it's gonna take time to reach everywhere. But within 3 months, this is the pace at which this virus spreads. So it's really, really transmissible. And once you have a variant which is fit, it's bound to spread widely quickly. Then we have the second variant of concern, which is the one which was selected in South Africa, first seen in October, now reported in 30 countries. And lastly, we have the variant of concern called variant 3P1. That's the one which was first detected in December and it's been seen in 8 countries. The color dark means that it's being seen in local transmission in those countries, while the purplish color indicates it's only been isolated in travelers. For instance, you can see the middle variant of concern, the South African one, the South African origin one has been seen in Kenya, but has been seen in travelers, while the B.1.1 has not been reported yet. So what defines these variants of concern? They are particularly variants of concern because they were seen to have very many mutations from what we expect to be the current clock-like rate of evolution of SARS-CoV-2. So the 3 of them have more than or close to 15 or close to that defining mutations. And surprisingly, 6 to 10 of these mutations for all of them occur in the spike protein. So it's mutation, but mutation which is much more focused on where we are targeting our interventions. And that makes them really important to keep an eye on. Of course, I've said there are other places where they have occurred, those differences. The open reading frame one, that's one of the areas where it codes for non-structural proteins. The spike protein you can see there for the variant 1, variant 2, variant 3. And one thing you notice for this variant 1, for instance, there is this mutation at position 501 from asparagine to tyrosine, and it's being seen in all the 3 variants, which made people think if 3 variants are rising independently and have a change at that position, must be an important position for the virus fitness. But also there are changes that are shared between the variant 2, the one now colored in blue, and variant 3. And one thing you can see, for instance, here we are saying this at position 18, having a change from lysine to phenylalanine, the same thing there. But you can see here, importantly, you have a change at position 417 from K to N, while here it has a change at the same position, but it's from K to T. So it's telling us that there are particular hotspots on the coronavirus which are prone to mutating. And there might be some advantages to that. So what are the risks due to these variants of concern? I like to just take you through some of the observed information, which is not yet peer reviewed, it's not yet thoroughly to scientific rigor. It has not gone through the process to be published, but it's what's the information that is out there. The first variant, that is the UK origin variant, information previously showed it was not, it was more transmissible, some up to about 70%. There is a confidence interval around there, 50%. So it seems to be attacking the population much faster. It has a higher attack rate than the other non-variant 1 virus which are circulating in the UK. It's actually become the dominant variant in much of the UK according to an FTAG report, which is the kind of the body concerned with advising the government there. The other thing is that initial information don't indicate it causes more severe disease, but a reanalysis of the data by 3 different groups in the UK has shown actually there is propensity to cause more severe disease. They have said that there is modest evidence, not kind of like it's absolute, like there is what they have a cautious name of probability, which they used to say, you can actually, there is evidence indicating that this variant could be causing more severe disease. And here they are measured, they have looked at people who are infected with this variant up to 28 days and seeing how many died versus those who were infected with the other variant. And they saw like there was up to 1.35 increased hazard ratio for those who are possessing this variant to die. And then finally, the other thing is that it's been observed to cause problems with diagnostics, what we call the S gene target dropout. So it impacts diagnostics, and in a way that now they are being forced to also have other additional ways of detecting the virus rather than relying on this alone. The second variant, which is the South African origin one, has been shown in at least 2 studies to have potential to escape neutralization from monoclonal antibodies, but for polyclonal antibodies, for convalescent sera, sometimes what it's shown that some people who suffered severe disease are more likely to have high antibody titers. They are less likely to escape this variant, but those who had milder disease, it's being shown like the variant actually can kind of have reduced neutralization, need higher antibody concentrations to be able to neutralize it well. There is no evidence that it causes more severe disease, this variant 2 from South African origin, but there is some evidence to suggest that it's more transmissible actually. Also, it's being indicated to up to around 50% more contagious. And the other information just for the variant 3, which is the Brazil origin variant, it's been associated with a devastating surge of cases in the Amazonas area, particularly the Manaus city, where in the first wave, they thought like they had the highest, one of the highest attack rate. They thought like they had reached herd immunity, but there is now a surge of cases, which is being driven by this variant. And that's why it's caused concern. And actually there have been documented cases of reinfection, but 2 are few still. But I think this is an area to just keep looking at. Just to also just add some good news rather than being a little bit more on the negative side, both Pfizer and Moderna have shown actually their vaccine does neutralize both the South African and the UK variant. However, Moderna said that they observed that there was, somebody said it was super awesome in neutralizing, now it's awesome. So it still does the job. So in a way, they have again tried to see if they can have a booster vaccine provided or constructed, which can be able to track these variants of concern, just to make sure that people stay protected for all possible variants. I do not say about the Kenyan epidemic, have that one, Lois showed that. So I'm gonna tell you what can we actually be able to understand about the epidemic using genomic surveillance. We are very much interested right from the beginning, given the measures of control we had in place, how many introductions actually occurred of SARS-CoV-2 into the country, and where were they from? We know we had sometimes targeted lockdowns, we had targeted in a way that we were allowing visitors to come in. So we like really to understand where are the variants which, the introductions which came in, and where did they come from and how many were they? When we are seeing an epidemic, for instance, Lois showed the first wave which had a peak and the second wave. We really want to know how many variants comprise those 2 different peaks and what were the transmission characteristics. For instance, is it that the second wave was driven by reopening of the economy, for instance, such that there were super spreading events, or actually was there so many variants in circulation such that it was inevitable we are going to have much more infections occurring. So variants circulating the country are quite of interest, as you can see, with variants of concern being spotted in various locations around the world. So we'd like to know whether which variants are in circulation and are they of concern, both in terms of understanding, is there anything that is Kenya distinct epidemiologically, immunologically, or in terms of causing more severe disease. And last but not least, definitely, we are using molecular diagnostics for almost all the cases we are reporting. Almost all are based on molecular assays, the PCR data. And this PCR data is targeting the genome, and the genome is, we know, is a moving target. So we'd like to know, do we need to update that? So to date, we have sequenced a total of 513 genomes in Kenya. And from those 513 genomes, most of them actually are from samples we diagnosed in our lab here at the coast. So they will be from Lamu, Kilifi, Kwale, Taita Taveta, Tana River, and Mombasa. However, we also did receive samples from Nairobi, from our colleagues there, and also from Kajiado. So I've just put all that data together as just Kenya data. And I'm looking here at how many variants actually have we observed so far. So we've seen a total of 29 variants, and here I call them lineages because that word sometimes is interchangeably used. But remember, variant is something which is just genetically distinct from another virus. One virus is genetically distinct from another, it's called a variant. But you can't call every mutation, you can't call it a variant. So once we have a class of virus with shared changes, we call them a lineage. So we have 29 lineages already detected in the country. And you can see they're not occurring the same in the same proportion. So you can see this particular lineage called B.1, which we saw right from the beginning of the pandemic in Kenya, has continued to cause infections throughout up to December when our data collection so far is. And you can see some months actually was even the only detectable lineage from our limited sample that we sequenced. Remember, Kenya has 100,000 cases all thereabouts, and we are only talking about 500 cases. Also important to say is that there are some lineages, for instance, the A, which you can see there, it's coming in, sometimes disappears. So this kind of pattern we are seeing, it's not unique to Kenya. Important to say, we are not seeing 29 new lineages in Kenya. We are seeing 29 lineages which have already been seen elsewhere in the world. I think it's important to clarify that. So this is what we can capture from our limited sequencing. And then if you look at those individual variants or lineages actually, which have provided there on the y-axis, and then here is across time, you can see the B.1, which I said dominated. So you can see we've seen it throughout. But there are some others which we just saw at the beginning, and you can see we've not seen them again. You can see some of them at the beginning. So this kind of, you can also give credit to our intervention. Some of them really were doing case tracking properly and making sure that people are isolated, makes transmission extinguished. But you can also see that introductions have continued to cut throughout. You can see some of them first seen there around June. You can see some of them first seen, for instance, that one there in July. That one there is the South African origin variant of concern, which we saw in 2 travelers. You can see there around December, and 2 cases. So you can see the whole thing about introductions are continuously happening, but also some of the strains seems to be disappearing from our radar. We don't know whether it's because of our sequencing or because generally they just disappeared. So if you look at the phylogeny of the sequences we've had so far from the 500 that I've just talked about, you can see their relationship, the B.1 you can see there. But importantly to say, which the B.1 there, which is this strain there in that particular color, which I can't say its name, you can see that they are actually not identical. So there is variation even within these lineages. And actually some of the variation is fairly huge. I think we need to kind of improve on our naming, in that some of these actually could be already lineages that we need to give names. Also importantly, you can see some of them like the B.1 3, you see it has a cluster, and when you track down, you find some of these clusters are county specific, meaning that there has been prolonged transmission within some settings, and that has led those particular places to have unique mutations. Important, this is based on the whole genome, 30,000 nucleotides, but what does it look like if you look at the spike region only? Spike region is about 4,000 nucleotides long. And the spike protein is where we said we target most of our interventions. You can see the diversity there is much limited. You see some of those which are called different lineages are actually identical at sequence level at that part of the genome. but some of them have that distinct difference. And I remember the South African origin strain you see in the spike, for instance, because of those several changes, it protrudes out. To conclusions, just to say there are variants of concern indeed, and they are spreading around the globe very fast. Definitely with our international borders open, there is risk that we will eventually see these strains locally. And the other thing is just to ask that it does seem like SARS-CoV-2 has some characteristic which is similar to what we have seen in influenza, which is technically called antigenic drift, whereby over time the virus moves away from what it used to be, and especially the proteins which are targeted by the immune response change their shapes and requires that the immune system continuously retrains itself on how to identify and neutralize this virus. So, and that has definitely implications. And that's why you see even Moderna already thinking about their vaccine having a booster dose developed, which is based on these variants of concern. And then lastly, so far the variant 2 and variant 2 have not been shown to be more lethal than other strains. But as I said, variant 1, the one that I'm sorry, I've called it there too. Variant 1, which was seen in the UK, has now been described as having a realistic possibility of causing more severe disease. Actually, that's the word I was looking for. That's how the UK experts call this, a realistic possibility. And then some words of caution, very, very important. With increased surveillance, we definitely are going to see more unusual genomic variants. And most of them actually mean nothing. They're just, the virus is just changed a little because there was that possibility. But a few, as you can see, I'm very sure that in the world at the moment, you have over 500 or over 1,000 variants, but 3 only have been brought to our attention. There is a concept called founder effect, where a variant can arrive in a place and cause expand the number of infected people. It's not necessarily fitter than others, but because it was the first to arrive there. So we need to be cautious on how, when we see more cases, even in the UK, there is no yet conclusive evidence that this is actually increased transmissibility. Actually, some scientists say it's hard to ascribe one change to increase number of cases because there is human behavior, inefficient lockdowns, increased testing. So there are several factors you have to take into account. But important also, our sampling protocols. If we are targeting, for instance, travelers, or we are targeting people in hospital, or we are targeting asymptomatic, our testing protocols, even our way we subsample those who we sequence, determines what we find. So it's important to take that into account. So some of the observations we are seeing are dependent on our surveillance protocols. And then finally, most of the data we are talking about is still in preprint, it's unpublished, and it's not yet peer-reviewed, meaning there is still scientific question that needs to occur to it before it can be accepted as new knowledge. What can we do? I think this should be my final slide. Simple interventions, as always, have the most effect or most impact. Before we wait for somebody to come down with a variant of concern, we definitely need to just adhere to things like just good hygiene, masking, social distancing, avoiding places that are crowded. And that does a lot even with the new variants. And definitely we need not to be complacent and just take stronger action and investigations anytime we see a concerning epidemiological situation. For instance, we are seeing a cluster of cases who are severely ill, and you are thinking like, wait, this is not normal. I think it's good to flag up and perhaps seek for assistance to see if we can well and put informed response. And finally, data collection is really important. How good data we collect determines what we see. So we really need to have improved data collection. It's always annoying to fill one more field, but that might be the field that is important for us to know how we need to respond. And finally, I have to acknowledge a huge team here in Kilifi. which has been responsible for running the genomics. I'm only presenting them today, and starting with Benjamin Sofa, who is our director, George Givinci, who is our really sequencing expert. He's been able to actually help us right from setting up to all the genomes assembly. That's really been good. Isabella, our head of department, and all the members on the slide, and also finally have to acknowledge James Knox and Philip John, who have really been of help as well in setting up this and helping us get to where we are. And finally, the work I'm presenting is funded by both the Wellcome and a grant which we got prior to the pandemic from the National Institute for Health Research, and it's from the UK. Thank you very much.
Thank you so much, Doc, for that excellent presentation. Sorry, someone needs to mute. Okay, thank you so much for that excellent presentation. I think you have broken down a complex situation into something very simple that you could actually understand. I think many people have struggled to understand what these variants are, and you're very grateful you could break that down for us. So I have a few questions sort of to help lead us into this discussion. One, what determines which variant becomes the predominant variant in any particular region? So maybe the question is, if we have people coming from the UK who are the variant, is it for sure that if we allow for transmission, then it will become the predominant one here as it has in the UK? Or what determines that? Sorry, please check your link again. We can't hear you. Sorry, Doc, we can barely hear you. Sorry, Doc, we can't hear you. Very poorly.
Okay, let me try again. Better.
But maybe you can put off your video and see if we'll hear you a little better, if the internet will support that better. Oh dear, now we can't hear you. Okay, what we will do, we're going to come back to this discussion. We can have just one other presentation, then we'll come back to you as you try and sort out the link issues, if that's okay. So what you can do for now is take down your presentation. To everybody else, please type your questions. We are going to come back to this discussion in a short while. Okay, please take, okay, thank you, Doc. All right. So we just want to cover something that's slightly different, and this is based on questions that have been presented to us severally over the last few months. And this is the issue of diagnostic challenges. Many times we have patients who come into the hospital with typical symptoms, typical image findings, yet their COVID-19 test is negative. And the question is, should we decide that they have COVID-19 based on the clinical and radiological results that we have? Should we isolate them? Should we manage them any differently? And here to help us answer this question is Dr. Nyukuri, who's a physician and working with us in the infectious disease unit at the Kenyatta National Hospital. Karibu, Dr. Nyukuri.
Thank you so much, Dr. Lois, for that introduction. This will be a very short presentation, and I'm very sure most of us managing patients in the COVID unit have had this diagnostic dilemma and wondering what to do next. Where do I go next? Okay, so I'll start with a brief case presentation. 49-year-old male patient referred from a peripheral facility. He was referred for ICU care, came in with a history of cough, difficulty in breathing and chest pains for 1 week that had progressively worsened over the last 2 days. At that peripheral facility, he had been newly diagnosed with diabetes and was also in DKA. So on examination, we found a middle-aged man on obvious respiratory distress, saturating at 70% on room air and 85% on oxygen by non-rebreather mask at 15 liters per minute. A few labs that he had with him, he had elevated WBCs with neutrophilia and left shift. Procal was about 1.2, which is almost 2 times. BGA, he was markedly acidotic. Bicarb was 8. Sorry, I can't remember the PO2, but it was also very low. RBS was 22. His COVID-19 PCR was negative. He also had a CT scan report that reported CORAD5, and CORAD5 is highly suggestive of COVID-19. I couldn't trace his CT scan report, so I borrowed something from somewhere. And as you can see, this is pretty much what a CORAD5 picture would look like. A bilateral lung involvement, there's consolidation. You can also see ground glass appearance, and mostly on the lower lung fields. And so we asked ourselves, so what do we do with this patient? Is this patient, does he have COVID-19? Is there something else? How are we going to manage this patient moving forward? So differential at that time, the patient, we came up with 3 differentials: severe COVID-19 pneumonia, possibility of an atypical pneumonia, and of course, he is a newly diagnosed diabetic patient in DKA. So I'll not go into depth about the management of his diabetes. Now, I came across this revised WHO case definition criteria for COVID-19 released in, I think, November, December last year. So a patient can either be a suspect, a probable case, or a confirmed case. So let's see where our patient falls. So is he a suspect case? A person who meets the clinical and epidemiological criteria is a suspect case. So clinical, again, as you can see, acute onset of fever and cough, which he had, in 3 of the following, which I'll not delve into. And epidemiological criteria resides in an area with high risk of transmission of the virus, or there's community transmission, or works in a healthcare setting. So we know at this time we still have community transmission of the virus. So I'll say the patient met the criteria for a suspect. Or if the patient has severe acute respiratory illness, which is defined as history of fever or measured fever more than 38 and a cough with onset the last 10 days and requires hospitalization. And lastly, if a patient is asymptomatic, does not meet the epidemiological criteria, but has a positive rapid antigen test, then that is also a suspect. Was he a probable case? Then this is a suspect who meets the suspect criteria, who meets the clinical criteria and has been in contact with a probable or confirmed case linked to a COVID-19 cluster, or a suspect with chest imaging showing findings suggestive of COVID-19. So our patient really did have a chest imaging showing findings suggestive of COVID-19, and we can say then he was a probable case. As he meet the confirmed case, the nucleic acid amplification test was negative, and we did not do an antigen test. So we can say this was a probable case of SARS-CoV-2 infection. Do we think the patient had a false negative PCR test? So what are some of the causes of a false negative PCR test? So if the patient had a low viral load, and we know early or late in the infection, the patient can have a low viral load. If there are sample collection errors, and you know, nasopharyngeal, and if you also collected from the nasal or mid-turbinate, probably have the highest detection rates. And also you need somebody who's well-trained in collecting these samples. The assay type that is being used, we know different assays do have different sensitivities. depending on their cutoff, the cyclic threshold cutoff, depending on the reagents that are used, depending on the manufacturers themselves, and so on. And from literature, I found that it can be anywhere between 5 to 40%. Do you think mutant viruses, as the previous presenter has mentioned, can impact the sensitivity of the test? As we saw, the S gene is the one that seems to be mostly affected, and there might be a drop-off in the detection of the S gene. But the good thing is most tests detect more than one target, some of them relatively preserved, and so this should not be a big worry. And if you see that the results are coming with, and the S gene is not being detected, then somebody can start thinking, is this a marker of variant mutants? Are we having quite a number of virus mutants in the population? And that's why the S gene is not being detected, and it's something that can be explored further. What about vaccination? Hopefully we'll roll out vaccination in the near future. Do you think it will have an effect on detection? So vaccination really has no effect on the nucleic acid amplification test, or even an antigen test. But if you decide to use an antibody test, which again is not recommended for diagnosis, then probably it can affect. So that should not be a worry. It's good to remind ourselves of this nice graph about, from exposure from exposure to infection, and the time at which the virus is highest in the body. As you can see, if we use an nasopharyngeal swab, from exposure to onset of symptoms can anywhere between 5 to 14 days, the incubation period. During around the time of onset of symptoms, that's the time when we do have the highest viral load, and in that week after onset of symptoms, that's when we tend to have the highest yield. So if the test was carried out earlier, then you can get a false positive. Then if you do the test after the second week, as I can see, there's a steep drop, especially in the nasopharyngeal PCR. However, if you're able to get a bronchoalveolar or a sputum sample, then it tends to stay positive for probably a longer duration. Antigen antibodies form after the first week. In the third week, IgM will fall in the fourth, fifth week, and the IgG will linger around for quite a while. So what do you do if the patient has a negative PCR, but you still have a high index of suspicion? So if you think that the test was conducted early, it's actually recommended that you repeat the test after 24 to 48 hours. And then you may get a positive test. What about the use of lower respiratory sample testing? Again, in hospitalized patients with lower respiratory tract illness, then yes, if you can get an expectorated sputum sample, or a tracheal aspirate or bronchoalveolar lavage in intubated patients, then that's something to be considered. However, it's not encouraged to induce sputum production. What about antigen testing? As you can see, if you go back to the WHO definition, antigen testing has a role. But in a patient with a positive antigen testing and meeting either the probable case definition or is a suspect and meets the criteria OB, then you can actually label this patient as a confirmed case of SARS-CoV-2. Also, asymptomatic patients with a SARS-CoV antigen positive who have been in contact with a probable or a confirmed case, then those can also be, those are actually also confirmed cases according to the WHO case definition. As I said, serology is not used for diagnosing COVID, it's more in surveillance. About the role of imaging. So chest X-ray may be normal in the early disease or in asymptomatic patients. In patients who are symptomatic, you may see hazy opacities when rounded in morphology. And also it will be bilateral and distributed in the lower lung. CT scan, it is more sensitive than a chest X-ray compared to nucleic acid amplification test, has a sensitivity of about 97. However, according to this study conducted in China, it had a very low specificity. And this is because there are quite a number of illnesses, especially viral illnesses, that do share the same presentation on CT scan. What is this presentation? You'll get multiple bilateral ground glass appearance, consolidation in the peripheries and in the lower lung distribution. Please note, no finding, no CT scan finding can completely rule in or rule out COVID-19. Okay, and therefore it's not recommended for screening or diagnosing COVID-19. It's actually something that is to be used to the management of hospitalized patients. So these are patients who whether is confirmed or you're suspecting, and you asking yourself what to do next. So that's where the CT scan comes in. Lung ultrasound, if you are able to, again in COVID-19 patients, then you'll see a thickened pleural lines and B lines. I don't know how many of us are trained to use it or have access to it. That's also an interesting discussion. So how will you manage these patients then? So as I said, the first thing, repeat testing after 24 to 48 hours. So our patient we repeated testing after 48 hours and it was still negative. Consider a lower respiratory sample if you're able to get. If the patient has a productive cough, that's a sample you can use. But also confirm with your lab if they're able to process a sputum sample, because most assays have been validated on nasopharyngeal swabs. So is this something that, you know, they can be able to give you a good results? The pre-test probability is very important. What, you know, does the patient have compatible clinical signs? What about the contact history? What about the travel history? So if your pre-test probability is high, then even with a negative result, you can go ahead and treat. If your pre-test probability is low, then you have to think about an alternative diagnosis. I remember being consulted about a patient who came in casualty. He had been involved in an RTA, was saturating in the low 60s and 50s. And on oxygen, I think it could not go more than 70. But this was very interesting. The patient was well prior to that. And after the RTA, when he was brought, that's when they found these findings. So I think that at that time that they wanted to know, you know, should we consider this a case or not? Clearly, you can see the pre-test probability there is low. I mean, even if the patient had COVID, it will be a red herring. It's not the cause of the hypoxia. It's most likely to be the RTA the patient was involved in. And I know there is a tendency for most patients, if we find a positive COVID or you're suspecting they have COVID, we forget about everything else and we concentrate on the COVID. So let's look at the pre-test probability. Imaging, we have talked about it, and it is to, you know, help us make a management decision. Always consider an alternative diagnosis, especially if the test results come back negative. So go back, look at the history, past medical history, and so. What about when to start and to end isolation? So looking at our patient, this patient started to display symptoms a week before admission. So even assuming that the patient was COVID positive, and we were to use the symptom-based criteria. So you can say, we know we are going to end isolation measures after 10 days since the onset of symptoms. and the patient is not febrile for the past 24 hours, and the symptoms have improved. Alternatively, if you have access to a test and the patient has 2 negative tests, really, then there you can consider ending isolation measures, and the patient can be managed in the general wards. Definitive management, again, as I said, it will depend on your pretest probability. If you feel that the pretest probability, the patient has COVID, you highly suspect the patient has COVID, then you can manage the patient as a COVID patient. And indeed, our patient, we managed him as a COVID patient. The good thing is he did well, and at the end of the day, he was discharged. I'll stop there and invite any questions. Thank you.
Thank you so much, Dr. Nyakuri, for this presentation. I think it really sheds light on what we should do with this sort of patients. Maybe just one question that I also see on the chat as well. How should we isolate these patients? How can they transmit if you're not able to detect SARS-CoV in the upper respiratory secretion?
Very good question, Dr. Lois. So if the patient, okay. So if we are not able to detect, we know patients are most infectious when they are shedding, their viral shedding is high. And if they're shedding a lot of viruses, you should be able to detect it because the PCR is very, very sensitive. So my take is, if you are not able to detect and you have done 2 tests more than 24 hours apart and the tests are negative, I think this patient can be managed just like any other patient. It's probable that the patient has completed, so if also look at the history. So if it is early in the infection, then the patient can be isolated, can be quarantined. You can treat the patient as a contact who has been exposed and need to be quarantined for the next 10 days or so, with a repeat testing at that time to confirm. But if it is late in the infection, and we do get the majority of these patients, you'll find that their symptoms started even 2 weeks ago, then it's most likely that these patients are actually outside the infective period and they can be managed in the general ward.
Thank you very much. And I think you really highlight some of the challenges we have when we have this kind of patients. But that was a very clear presentation. Many thanks. Dr. Charles, have you been able to link up again?
Yep, I've been able to link back.
Excellent. So I'll just repeat the question I'd asked, and it sort of ties in with some of the questions that have been asked on the Q&A platform. So one was, what determines which variant becomes the predominant sort of circulating variant, or the one causing local transmission in an area? If we have 1 or 2 cases that have been identified in country, how concerned should we be? And I guess it ties in with one of the questions I see from Dr. Kibias. Does that mean, should we ban flights from the UK and South Africa, or people coming in from Brazil? How do we navigate those questions?
Thank you, Dr. Lois. It's really interesting question. I think about dominance, I'll say like the variants have an open competition against each other. And normally what happens actually, then properties of the variants become important, and properties of the host as well, or behavior of the host, as they say, becomes important. So one, there is the aspect of founder effect. So which variants occurred in an area first normally has some impact on which variants are going to be dominant. But also as time goes by, this development of particular mutations in some of them, which perhaps help the variants to bind better to the host receptors, to replicate better, maybe to avoid some low level immunity in the host, that really also promotes dominance of some strains over others. But having said that, remember also there are aspects like if you have variants circulating in a place where there is no lockdown, and those were the founder viruses there. For variants which are fit, which are circulating in a place where there is a lockdown, there is extreme contact tracing, and all kind of measures. For instance, what you see in places like Australia, where there is even lockdown between states, then you are going to have very limited circulation of even fit variants. So I have to say that overall, it's dependent on multiple factors beyond just the virus alone, beyond the host alone. In terms of, should we consider banning flights from South Africa, UK? I think no. If we were to do that, we were to do that really early on. And really early on, very few people knew these variants are going to spread this widely. The reason I'm saying this is because you've seen those variants, for instance, in the one in the UK, it's already been seen in 64 countries. So if you ban flights from UK, what about the other 63 countries it's been seen? The same thing to the South African variant. And remember, we are not having evidence of absence of those variants in many countries, including our own, because there is this statement that goes absence of evidence, not evidence of absence. So I think at the moment we should much more focus on understanding better what's circulating within and just taking on board what we know already works, the tools within our reach, which are so cheap, and yet it's very difficult for us to adhere to them, social distancing and things like that. So that's my take.
Thank you so much. Sorry, go ahead.
Just to answer the question that follows, actually, that about the R0 of the new variant, the same thing happens. The R0 of the new variant, or the so-called reproductive number, is dependent on those several factors. For instance, a variant circulating in a place with a lockdown is likely to have a lower R0 compared to a variant circulating in a place without a lockdown. So the statement that they are about 50% more contagious is a statement that they have higher R0 than previous variants. And the UK, initially they said it's like 0.04 to 0.07 higher than what was circulating there.
All right, thank you very much. I have one other question for you. After that, I hope Prof. Nzala is here because I'd like you to answer a question for us. For Dr. Nyaigoti, I just wonder, learning the lessons we have from other viral infections, let's say if we borrow from HIV, for example, even though we know there are changes that occur with time just because a virus has to be a virus, we know that certain mutations may be triggered by use of partially effective antivirals. So for example, if I have a partially infected antiretroviral, then I may trigger resistance to other antiretrovirals because of certain mutations. As the world struggles to get some treatment for COVID, we may have used a number of partially effective antivirals and we may still be testing a few. How much do you think the use of partially effective antivirals against COVID-19 leads to development of certain mutations that may lead to variants of concern? And this is the same case with use of monoclonal antibodies and maybe particular vaccines as well. Is there any work around here, and what are your thoughts on this?
That's really, really good question. Thank you. It just goes really in terms of, it's a hypothetical situation whereby if you really need applying some pressure on the virus, but that pressure is not to the level of eliminating the virus from the body, that means you're going to have that sort of prolonged infection period. And that's normally the window when the virus has opportunity to have developed some mutations which might be actually overcome the pressure you are applying. And I agree, and it's a hot scientific research topic at the moment, trying to understand. For instance, in the UK, there was this debate giving one dose versus giving 2 doses. So how will that actually relate to a population which is kind of semi-immune, but that pressure, that window that still is left, that probably the virus can be able to still infect some of these vaccinated people. It's important, as you've said, actually, the monoclonal antibodies. This, even in vitro, if you grow the virus in the presence of monoclonal antibodies at suboptimal concentrations, the virus develops a mutation that makes that monoclonal antibody not to bind. So actually this is a universal, I'll say, sort of application to many of the viruses. HIV replicates 4 times faster, as I said, than SARS-CoV-2, so has better potential to develop resistance compared to SARS-CoV-2. But as you can already see, even the UK variant, scientists on observing, they said this must have come from an immunocompromised individual, and most likely who had a prolonged infection, and this is where we are having that challenge of suboptimal tackling of the virus. In terms of what we can do, I think, first of all, developing an understanding around the area is the first thing to do, because you are able to address better what you understand. So we have local patients locally who we know they have been infected for a prolonged period. We know what they have been on. We know some of them, how responsive they have been to our medication or our treatment. Being able to go the extra step to characterize that, and that's why I say data makes us know what's really working, what's really not working, and what's becoming a problem than a solution.
Thank you so much for that excellent response. And I think maybe for all the people listening, it's important that we use effective therapies and to avoid things that have very little evidence for use, because as I said, there's this potential, theoretical potential, that when we use things that are partially effective, we could lead to driving certain mutations. So again, let's use what is evidence-based. We talk a lot about treatment. I would revisit the issue of treatment to debunk some of the myths around some drugs that may potentially be used. So I have a question for Professor Nzala. There's a question that's been asked here around vaccines, but actually my question is around reinfection. We've seen what's happening in Brazil. The Manaus area has had a very large spike. But as of last year, we were led to believe that most of the Brazilian population had already been infected. So what is it about this new surge? Is it that there was no herd immunity developed with infecting a large population in that context? Or is there something about this variant that may escape the immunity that was already developed, if any, to the initial infection?
Okay, yeah, okay. Thank you, Lois, for the question. But I also like to thank Duncan and also thank Charles for the very good presentation. But before I talk about immunity, just give me a minute or so to also give a sense to the question you first asked about subtypes and issues of dominance to Charles. I'll just like to bring to your attention, ladies and gentlemen, that we have had HIV for over 30 years. And if you look at HIV and you look at subtypes, Southern Africa has remained predominantly subtype C. Kenya, Uganda has remained predominantly between A and D. Ethiopia here is predominantly subtype C. So we've struggled with that aspect, that how can HIV over 30 years, and we saw mixing of individuals back and forth, regions still remain predominantly with certain subtypes. Even in Europe and North America is subtype B. We've been looking for subtype B in East Africa. We only saw a glimpse in the Mount Kilimanjaro area some time back. We don't see subtype Bs. So that question, Lois, is a question that we are still working on, just to understand what really happens. But what we think could be part of it could be that even if the receptors are the same, there could be receptor heterogeneity in the various regions. And then number two, there could be an element of host genetics. And this is an area of interest, ladies and gentlemen, where we are beginning to ask ourselves, how does the African genome influence susceptibility to infections, disease progression, and outcome? So we are many in this group, but these are the directions we really want to take in terms of unpacking, those aspects. And then the last thing could just be the immune environment in which we find ourselves. So as you talk about these variants, as you talk about these subtypes, as you talk about dominance, reflect on really what is going on HIV, which has been with us 30 years. We hardly see subtype B in Kenya. We hardly see subtype C in Kenya, and yet just to the north here, Ethiopia is predominantly C, and yet cross-border movement is always there. So I just wanted to bring that up so that I highlight these issues of host genetics and the susceptibility to infection, issues to do with the immune environment, and more so mucosal environment and innate immunity in that aspect, because these infections tend to be mucosal. Now, turning back on your question, Lois, on immunity, it's now clear. Last week, we had a discussion with some colleagues, and there is definitely an element that the immunity that we get from COVID is three aspects of it. There's immunity at innate level, innate immunity. There's immunity through B cells, which is giving you antibodies, and there's immunity through T cells. So these are the three aspects. So the question we are asking ourselves, post-recovery, which of these plays a big role? That is a real question that we have not clearly unpacked, because it will need studies that we are engaged in now as we are collecting more samples. But what we have also very, very clear is that the B cell immunity begins to wane very rapidly. In some people, post-infection, antibodies are only lasting between 3 at the most to 3 to 6 months. But even if there are no antibodies, a question we are asking, can innate immunity protect you from reinfection? Or can T cells protect you from reinfection? So reinfection is taking place. That is already clear. And even in Kenya here, maybe we are not looking enough. Reinfection is actually, taking place. The immunity triggered, the immune response that is triggered by B cell could be slightly different from the T cell, and also could be slightly different from the innate immunity. But it is now clear the B cell immune response maybe last 3 to 6 months at the most. Reinfection is actually taking place. So the studies that Charles are doing in terms of looking at COVID sequences needs to be expanded to relate not only to sequences, but also to relate sequences to symptomatology, so that we know these sequences are the kind of sequences we are seeing in individuals who are asymptomatic. These are the kind of sequences we are seeing in individuals who have mild to moderate disease. These are the sequences we are seeing in those who have severe disease. And now we are also sending child samples to look at sequences in those individuals who passed away. So I think where you sit, Lois, help us to be able to begin to unpack with reinfections. We see these infections, we are now in a position in Brazil is that might not be able to actually give us a herd immunity that we thought about, because maybe the immunological response does not through natural infection. And that's why maybe vaccines would have. So I'll stop there. Thanks.
Thank you so much, Professor Nzala, for simplifying that for us. I think recently there's been some data, not peer-reviewed as yet, suggesting that even in South Africa, when they look at sera from patients who previously had COVID-19, it doesn't seem to neutralize the new variants, where they have the variant 2. But then again, as you say, it may not just be about neutralization. We need to see how the other aspects of the immune system are coming to play. We're almost done. There may be one or two other questions. One of the questions that's been asked, there's been a few questions around vaccines, and maybe we'll have another session just to look at vaccines again soon, just before hopefully they arrive and are rolled out. But someone asked, in a country like Israel, where giving a single dose to the elderly has seen a drop in 60% in hospitalization, should we give a second dose or just give the others to everyone else? And I think it goes back to something that we mentioned earlier, and Charles said weighed in on this. If we give partial immunity, do we potentially drive mutations and new variants? So you may then end up reducing that current outbreak, but then you have another one with variants that may not be protected by that particular immune system. So the recommendation as of now is that you give the vaccine as intended, with the 2 doses. And I think even the countries like the UK may be rethinking their strategy of giving a single dose first. But of course, issues of availability of vaccines will continue to be a big discussion as time goes on. I think, Dr. Nyukuri, there's a question for you. I'm trying to find it. Oh, it's been answered by Prof. It was around the vaccines, yeah?
Yeah, Lois, just to add on what Nyukuri presented, you see now, and I've always I've been an advocate of colleagues in infectious disease, that the patients who fit case definition for COVID, what else should we be looking for? In that sample that you send us at the lab, what else should we be looking for? Because when Nyukuri presented, what we would have also liked to see is what are the alternatives? What else could this patient be having that could actually present in this manner, even if they presented in early enough and there's no COVID? Because what I'm thinking of is that we can develop platforms, and Charles will agree with me, of multiplex PCR, where you tell us that when this sample is coming, in addition to COVID, look for these other viruses that are likely to also present in the same way, so that it is now easier to quickly say this is not COVID. I think we think this is this. Then you can actually move that patient to another level of treatment. So for me, let this discussion help us also lead to practice. So I'd like Duncan to just comment on that. What else could actually, this be? And I've asked myself very many, hard questions. And I know Ruth Ndwat is here. Is RSV only limited to children? So Duncan, I want to hear from you, Ruth Ndwati, as well as Lois. Is RSV limited to only children?
Thank you very much, Prof. Maybe I'll take the liberty of answering on behalf of the others. In other hospitals where we've been able to look at the respiratory biofire, even in patients who presented with typical symptoms, looking at presence of other viruses. So for example, the biofire would look at the presence of influenza rhinovirus, parapneumovirus, respiratory syncytial virus, metapneumovirus, and others. In adults, mainly the ones who present with severe disease, often you'd find maybe if it's flu. We haven't seen a lot of flu since the outset of COVID, but previously, most of the time, what we find causing severe pneumonia would be flu. Occasionally, you have patients presenting with severe pneumonia, and maybe what you isolate is a metapneumovirus, which is often of concern to us in patients who are immunosuppressed, particularly. In terms of RSV, I think we can look at data from facilities that have routinely done the respiratory biofire or other multiplex PCRs on respiratory samples to see how much of it we've seen in adults. In practice, I haven't seen that many, but we can also talk to colleagues from other hospitals to see what they might have picked, and that's restricted to adults. But I totally agree with your comment. We should not just assume that because we're in the midst of COVID, everything that smells and breathes like COVID is COVID. And we really need to look for further support to, investigate these patients further and look at some of the multiplex PCR on their respiratory samples. It will help us see if there's anything we are missing. As Dr. Nyukuri presented, over the last few months, we've had a lot of patients presenting very severe respiratory illness, whose CT scans are typical, with that Corad's
Hello, Lois, I can't hear you, or is it just me alone?
You can't hear?
Yeah, I couldn't hear you.
I'm sorry. So I've just been talking to myself and Nyukuri and Thiranira over here. So I'm saying over the last few months, we've had a lot of patients who presented very typically like COVID, in whom we looked, even including having lower respiratory samples and looking for other potential infections, and we did not find any. So in the context of this outbreak, we have managed them as COVID. But I think this is work that needs to continue so that we know for sure whether there's something else that we may be missing. Sorry? Prof. Ndwati, you have a comment?
Okay, so Lois, thanks a lot for for that, because for me really, these are great discussions we are having with colleagues, and we must build on this in terms of improving the practice. So as the next thing that I'd also like to ask is, if COVID is going to become endemic, influenza, we know influenza because of the issues of zoonotics, that these things are in the wild. But if COVID is going to become endemic, in what form will it be? Is there a possibility that there could be a level of carriage? And Charles, I would like you to talk to this a little bit, because SARS-CoV-1, if I may call it so, disappeared. MERS-CoV is in northeastern, but it is kept and reservoired in camels, and it infects humans. But if, as I see that this virus is tending towards endemicity, in what form, Charles, do you think that this virus is going to actually exist? Over.
Thank you, Professor Ansala, and thank you for your earlier clarification about receptor heterogeneity and host genetics. I really liked that explanation. So in terms of potential for endemicity of SARS-CoV-2, I think that's real, especially given the sorts of mutations we are seeing, which sometime, as you've seen, the risk actually, some of them, the neutralization from using previously developed antibodies is much lower, although as you again explained, the other arms of the immune system, we've seen a bit of reinfection. And if you think about this and think about influenza, which behaves fairly similarly, where then the disease becomes quite edged or pushed to particular population groups. For instance, influenza, as we know very well, we have at-risk groups, especially the over 65, and sometimes in some cases, the under 5s who are yet to have their own exposure to develop immune responses towards the virus. So there's going to be a number of players in terms of driving the endemicity. And importantly, as you said, actually, we are seeing I don't know whether to call it carriage. I don't like calling viral carriage. I like calling asymptomatic infection, but because ideally we should clear viruses when they come into our bodies using our innate mechanism T-cell responses, we should clear them. But we think people abroad who are infected up to 150 days, those are 5 months of carrying the infection, meaning it's possible actually somebody can carry the infection through a period till the antibodies in already vaccinated or previously exposed people, those antibodies to went to a level that they can again get infected on re-exposure. So, Prof, I know you understand this, that it's a difficult question without the benefit of time seeing what happens 2 years post-emergency, but there is every indication this is going to become an endemic infection.
Exactly. So I think the big thing is that we must just begin to design studies to look into these and then prepare ourselves for the long haul. And even in addition to that, have a constant surveillance system where the vaccines will be made more or less just the way we are making influenza vaccines. Over.
Thank you very much,
Prof,
for that discussion. And thanks, Charles, for the response. I think Prof. Ndwati had one comment. So maybe if you could just take a minute or two, Prof. Ndwati, then we can continue.
Yes, thank you very much, Lois. And first of all, thank you to the panelists. This has been a really excellent presentation. I was just making an observation of a case I recently was connected with of an adult, actually a health worker, being ill, mainly a GI kind of symptom, nausea, vomiting, a bit of a runny stomach, and at that time testing negative for COVID using the PCR testing. And then during convalescence, the repeat test during convalescence actually being positive, and clear evidence of transmission to a close family member. So I think what Lois, you said earlier, that when you have somebody who meets the criteria, the clinical criteria, in the middle of this pandemic, we really must consider them to be probably COVID infected and do the needful in terms of preventing infection to others. So, for example, self-quarantine, all the usual things that we are recommending. Thank you. That's my only observation, and thank you for the excellent presentation.
Thank you so much, Professor Ndwati, for that observation. I think it's very valid because we have seen situations like that where two people come in with very similar symptoms from one household, one tests negative and the other one goes ahead to die positive with COVID. So again, in the midst of this context, we continue to treat as appropriate until we have alternative answers. Thank you so much. I think there's just one other question that was asked around the issue of this recent publication by Kemri Wellcome Trust around the seroprevalence study. No, that was CDC, around the seroprevalence study that showed that quite a number of people had potentially already been exposed, that our detection rate is extremely low, but also our mortality rates, if we take into context the wider infections, including those that had not been diagnosed, would be low. So one good news that our mortality rate would be quite low if we take, everybody, including those with asymptomatic infections. We also know that in the context, especially towards the end of last year, there are very many people who had mild symptoms and stayed at home and never got tested. So we truly do not get a good sense of how many people are infected. What this means for us going forward, I think, is anyone's guess. From the situation we're seeing in other countries and from the discussion we have had, being seropositive does not necessarily confer absolute immunity to future infection. So as Dr. Charles very well put it, we still have to continue adhering to the basic measures of mask wearing, hand hygiene, and avoiding gatherings until we understand this better. So thank you all for your attention, for the questions, for the discussion. A special thanks to Dr. Charles. It's first time joining this webinar, and you've given us an excellent presentation. We will continue to get questions. You can send in your questions even during the week. We try to focus our discussion on the questions that we are receiving from all of you. And we'll have our next webinar in 2 weeks' time. Have a good weekend, everybody. Or in 1 week. Dr. Thuranira says next week. Okay, you will be informed for next week. Thank you so much, everyone. Have a lovely, lovely weekend. Bye.
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 Okay, bye-bye.
Thank you
Thank
very
you.
much.
Yeah.
Bye-bye.